In the early 1900s, this fellow right here, Paul Ehrlich, noted that although the immune system is designed to protect us, it can sometimes go awry and attack uh, us or self. He termed this as horror autotoxicity, or fear of self-poisoning, and later it was termed autoimmune disease. And those autoimmune diseases are defined as autoimmune diseases are disorders in which immune responses are targeted towards self-antigens and result in damage to organs and tissues in the body. This can be from T-cell mediated or autoantibody uh, reaction to the host. 5% of the world's population, and they are quite common. We do a lot of autoimmune testing in the laboratory. I'm not going to go through every single learning objective since we go through them in detail in the lecture. However, once you've seen the lecture, go back and look at the learning objectives and see if that we've accomplished all those goals. And if you haven't, go back and read that specific slide. So how does this all happen? In earlier lectures, we talked about the fact that the immune system can recognize self or non-self. Non-self initiates the immune response. So self-tolerance is the ability of the immune system to accept self-antigens and not initiate an immune response. Autoimmune diseases are obviously the loss of such ability, and so self-tolerance is a type of immunologic tolerance. In other words, it tolerates an antigen that it finds and does not initiate the immune response. Lymphocytes have to be educated so they can distinguish self and foreign antigens, and this education takes place, as we know, uh, for the T cells in the thymus, and B cells go through a different maturation process, but the T cells get a random antigenic markers put on them in the maturation process. And so, since that process is random, certain antibodies are going to be developed just by chance that are autoreactive. And uh, by a process known as central tolerance or negative selection, we eliminate those antibodies that could have a destructive effect or an auto affinity to self. Uh, the T cells with less affinity for self, uh, which migrate out to the lymph nodes, uh, will maintain this tolerance, and then there's another process, the peripheral tolerance, that deals with that. Positive selection is the process that ensures that the mature T cells recognize non-self peptide MHC complexes. So that's what they're supposed to do, isn't it? So that was the process that took place in the thymus. Once they go out into the world, so to speak, and they find themselves in the peripheral lymphoid tissues, there's a peripheral tolerance that can happen in several ways. Uh, we talked about in the maturation process of antibodies in earlier chapters the way that the peripheral tissue can delete self antibodies through apoptosis. We can make the T cell unresponsive to antigenic stimulation through energy, or we can go through the T regulatory cells, which can inhibit the function of the T cell to do what it was supposed to do in the first place, but we've mitigated it through that control mechanism. So, as with the T cells learning to be self-tolerant, the B cells go through a similar process. We know that the T cells are named T because they mature in the thymus, and the B cells, the bone marrow. Uh, so, with the B cells, we have to go through the same type of process, but in this case, B cell tolerance involves apoptosis, but of the receptor. And this will kind of delete the receptor or the particular kind of antibody that that B cell can manufacture once it becomes a plasma cell, and change the genes, rearrange them, and result in a new specificity for a new antibody. In other words, it becomes no longer a self antibody. And in a similar way to the T regulatory cell tolerance, the peripheral B cell tolerance still retains the and encounters the self antigen, but in the absence of the T helper cells, they become either unresponsive or they die due to apoptosis. So, with that knowledge and the knowledge of how the antibodies develop in our body and how we go through a safety mechanism in order to uh, figure out whether it's a self antibody and remove it or mitigate it. There are instances where you can go through this entire process and still end up making an autoantibody. And how that happens is not really perfectly known, but it does seem to run in some families, and it is associated with exposure to certain environmental toxins and such. So, how a particular person ends up with an autoantibody disease, it's kind of a family thing or a toxin thing, not really sure. But from our perspective, uh, that matters little because we're really in the laboratory in the business of uh, trying to determine the specificity or uh, presence of autoantibodies. This slide just goes into a little bit more detail about that. You can have a genetic background that makes you more predisposed to an autoimmune disease, and that has to do with the makeup of your own MHC complex and whether your MHC could have a genetic or HLA makeup that makes you a little bit more predisposed to having an autoimmune disease. What do I mean by that? Because if your HLA expression is similar to a bacteria, say, it's possible that there could be some crossover or something like that. This diagram is in your textbook, and it basically shows the factors that go into it, the predisposing factors. Uh, it actually makes it through the immunological tolerance self check, and you end up with an autoimmune. So, there's several ways that this can happen, and we're going to go through, I think, about six of them. Molecular mimicry. This is when you're developing an antibody to something, but uh, that antibody resembles or has epitopes that will attract possibly antigens that are self. They're close enough where you can get a antibody antigen reaction and kick it off. This is called molecular mimicry. The second one is the polyclonal lymphocyte activation. This can happen in CMV, Epstein-Barr, due to lectins, and they can activate our B and T cells, but they're not specific. They just cause damage. Third is antigen spreading, and that's when you have a process going on throughout a disease where the inflammatory process is recruiting the T and B cells repeatedly, and as a process that can be some cross reaction, and then that usually ends up with multiple self antigens being recognized, not just one. Sequestered antigens. One of the ones that pops up with sequestered antigens is, say, the lens of your eye. There are certain proteins that are hidden from your immune system because of the nature of that protein being kind of cryptic or hidden in the tissues of the host. So the blood antibodies don't really have much circulation or exposure to that. But if you get injured, and say the protein to your eyelid gets released into your bloodstream, this can then cause or initiate an immune response, and you end up with an autoimmune disease. I'm not going to test on this particularly too heavy, but hormonal influences can play a factor. I don't really think this kind of almost falls under the idiopathic. It might or it may not. So hormonal factors can play into autoimmunity. Certain hydrofluorocarbons, other toxins in the environment can change the nature of the protein marker. This doesn't necessarily mean that you're changing the genotype of the individual, but the expression of the protein gets changed. And once it gets changed, it can be recognized as foreign and again initiate the immune response. Here we just have a breakdown of everything that we discussed. Probably a good study slide for you guys. So how do we determine a diagnosis of an autoimmune disease? And that's done in the laboratory. And from our perspective, it's usually the presence of autoantibodies. In previous lectures, we discussed what the ANA is, the anti-nuclear antibody, and we saw examples of the various patterns produced by these autoantibodies on the cell because they are particular to a cell organelle, and we use a fluorochrome that finds that's attached to anti-human globulin. It will seek out specifically and geographically in the morphology where the specificity of that antibody is, producing a pattern. When we're talking about ANA, we're talking primarily about SLE or systemic lupus, but there are more than 100 autoimmune diseases, and we break them down into categories. Systemic versus organ specific. We're going to go over some of the heavy hitters in the systemic autoimmune diseases, and then we'll get into the organ specific. You'll see under the organ specific category that because it's organ specific, we tend to kind of study them with regard to the autoantibody specificity that it targets. Here we have a breakdown You see systemic lupus, rheumatoid arthritis, and glomerulomatosis, and the associated autoantibody with it. This chart will probably make a little bit more sense as we get further into the lecture. So, lupus. So the first of the systemic autoimmune diseases we'll talk about is lupus. Affects women far more than men. And for us, the double-stranded DNA is the biggie antigen. There are several antibodies involved, other nuclear components, lymphocytes, RBCs, and go on down the line. You'll see that once we get into the ANA itself chart for the antibodies. But double-stranded DNA is one that would be the primary one associated with it, and probably the one that would be in the testing on the exam. It also has cell-mediated immune response involved and can attack the kidney. It's systemic. So you have joint involvement, skin rashes, renal, neurological problems, and so on. So how do we find out if someone has systemic lupus? Your CBC, and this is a little bit unfair to include this at this juncture, but when you go back and you've had hematology and chemistry and urinalysis, these will make a little bit more sense. So there is a little bit of backtracking involved in medtech, but suffice it to say that the evaluation of a CBC can help differentiate but not diagnose. The ANA is typically the screening test that we rely on for systemic lupus. As I previously mentioned, the chart for the ANA is extensive. And as we looked at the patterns of the ANA in class that the fluorochromes produce under the microscope, you see that you have homogeneous, fine, coarse, but if you look to the left, there's more than one antibody by far involved in systemic lupus. As far as examination goes, you're not going to have to memorize every single antibody here, but you would be responsible for understanding the fact that if a positive ANA is detected, the pattern can be reversed back to the particular type of antibody. Of course, each test for the ANA would be using an individual autoantibody, so we can determine at the end the spectrum of antibodies that that patient may be positive for or not. Most of these antibodies for systemic lupus are targeted towards cell nuclei components. Obviously, the big one is the double-stranded DNA. Single-stranded DNA, histones, and the like are also included. So how do we detect them? The big one is the indirect immunofluorescence test, and that is that's the visual one rather, that we will be focusing on, although in your ANA, under ANA in your textbook and in your ANA video, we mentioned the various other immunological techniques that we covered in earlier chapters can also be applied, because anything that's utilizing an antibody or a specific antibody against double-stranded DNA, say, all of these methods can be employed because they all rely on a single specificity, don't they? So here we go. Indirect immunofluorescence. We all know that we're going to put a dilution of the patient's serum onto the slide, which contains a human cell. We're going to allow any such antibodies to attach specifically, geographically, and morphologically onto that cell. We'll then wash the assay, and if it's positive, we can dilute that serum out to determine titer. And that's really what we're speaking about here. If you don't have your serial dilutions down by now, please go back to that chapter and review, because it will bite you when we're speaking about positive DNA. So again, let me get my pencil here. As you see, we have the cell has already on it the human cells are affixed to the cell. This little bubble right here is representing putting the patient serum on, and if any antibodies happen to be present in said sample, they will attach to the slide. However, they are not visible at the moment. In order to do that, we need the anti-human globulin, which has a fluorochrome attached to it, which will emit light back to our eye when excited by a laser beam. And if indeed it is positive, it will be geographic in nature, meaning that it is attached to the specific organelle or location within the cell that that antibody has affinity for, resulting in various patterns. The homogeneous pattern is fairly self-explanatory. The entire cell is glowing. Rim, speckled, nuclear, and centromere should be rather obvious from your anatomy and physiology, but they represent the specific cellular organelles and components that the antibody has a specificity for. These patterns are characterized here in the next few slides. I'm not going to take the time to read them all to you, but suffice to say that you will be responsible for these patterns and the description of the pattern in the slides here. But if you refer back to the previous diagram, I think it's fairly self-explanatory. Speckled, nuclear, centromere. And here you go. Here's some examples of it. You can see here in the first one that we have a homogeneous pattern, the largest picture here, and the entire cell is glowing green. Some mixed patterns can be observed. I did ANA testing for a little while, but this is something that becomes more of an art rather than a science, and takes a little bit of experience and training on the bench to develop. Another side note here is, although the ANA is considered the test for systemic lupus, it does have a false negative rate of 5%, so it cannot completely rule it out. The next systemic autoimmune disease we're going to talk about is rheumatoid arthritis, which causes a chronic arthritis in the peripheral joints. Again, systemic, and it results in pain in the joints and deformity, and also has a number of additional sequelae that can develop: subcutaneous knowles, pericarditis, and the like. Uh, in this case, we have autoimmune antibodies form with antigens to form immune complex. This is going to cause an immune response that leads to inflammation and cell-mediated response that will start to cause lesions and destroy tissue. This is the primary thing that happens. Two antibodies involved, but primarily the rheumatoid factor. What is rheumatoid factor? It is an autoantibody, usually an IgM, the larger of the immunoglobulins, and it reacts with the FC portion of the immunoglobulin G. It is found in 80% of the patients, but it's not specific. The next one is the anti-CCP, cyclic citrulated peptide. ANAs and ESRs, CRPs, and complement can be involved. The ANA and the ESR, ANAs were covered, but ESRs will be covered in hematology. I really am not going to test you on that. I don't think it's fair because you don't know what that test is. Okay. Rare granulomatosis. Having a little bit of trouble. Wegener's granulomatosis, which is a rare autoimmune disease involving the small to medium-sized blood vessels. Most patients will have antibodies to neutrophil cytoplasmic antigens, and this results in damage. Hence the word granulomatosis. This is getting kind of into organ specific. And again, as was mentioned, these are anti-neutrophilic cytoplasmic antibodies, and they are directed against cytoplasmic granules within the granulocyte. Same kind of story here as the ANA, as we can detect it by immunofluorescence on a slide of the leukocytes. So you would see that pattern geographically, morphologically, in that technique, whereas the ELISA or other methods would just come up with a positive and negative. We can apply all those immunological techniques again to that, but the fact that the neutrophils are glowing in place is somewhat helpful. And again, a chart that shows you the appearance or the pattern and what you end up with. You end up with a diffuse pattern or the lobes themselves glowing, and that'll determine the type of positive you have, and hence the specificity of the antibody. An example here we see them. This is a homogeneous pattern, right? And in this case, we see the components glowing. Okay. Now we're going to get into a series of diseases that are organ specific. And if you notice, all these charts are in your textbook in the chapter. But if you notice, for Addison's disease going on down the line, you have a antibody that is specific to the type of cells within said organ and causes a specific disease. These organ specific autoantibody diseases, I will require you to know all of the antibodies for, because the specificity of the antibody is really the pathology that causes the problem we're talking about, versus systemic lupus, where there's 10 antibodies, but we'll focus primarily on double-stranded for that one. But it's a systemic disease with multiple antibodies. This is a specific organ disease, and you want to know the type of antibody involved. So we'll go through that. Again, the chart. Autoimmune thyroid disease. I hope everybody remembers from anatomy and physiology, TSH is the thyroid stimulating hormone, and there's a feedback loop involved in with the pituitary gland to produce T3 and T4, which regulate your metabolism. So, if you have an autoantibody that's produced in the process here, either in the negative feedback loop or in the production, you can move them from hyper to hypo, depending on the specificity of that autoimmune disease and where it has a problem in the process. Hashimoto's thyroiditis, inflammation of the thyroid, results in a goiter, additional problems, dry skin, weight gain, as you would expect, because we end up with a hypothyroid situation, which means low T4. And if you have low T4, then it would make sense that you tend to have a higher thyroid stimulating hormone in place, trying to kickstart or increase T4. Antithyroid peroxidase antibodies and antithyroid globulin are present. Graves' disease. Uh, you fellows might be a little bit, your folks might be a little bit too young to remember Don Knotts, but he was a actor that had this kind of bug-eyed condition. He produces this particular uh, exalamos, I believe is how it's produced. Um, but it's thyroid stimulating hormone receptor antibody, results in low TSH, high T4, which means the person is hyperthyroid. Their metabolism is ramped up too much. Type 1 diabetes. Type 1 diabetes is caused by the destruction of the Langerhans cells in your pancreas, uh, which results in an insulin deficiency. The laboratory results, and you haven't had hemoglobin A1C in chemistry, I understand, but you will obviously have a higher glucose level, resulting in a higher glycolated hemoglobin. Questions on that I can answer in class. There are those are the specific autoantibodies involved against the Langerhans cells. Moving on. Celiac disease. This is organ specific to the small intestine, and it's triggered by antibodies that develop to gluten, and therefore you end up with a autoimmune response that's damaging the intestinal tissue, uh, and the antibodies involved are listed here. All right, immune-mediated liver diseases. When you have damage to your liver, you're going to have abdominal pain and elevated liver enzymes, and the antibodies are listed there. Multiple sclerosis, autoimmune disease that involves inflammation and destruction of the central nervous system, affects the brain and spinal cord, and you have antibodies that are specific to that. I don't believe I have the antibody listed here, and I will get back to you guys on that. Myasthenia gravis, neurotransmitter antibody. Um, This will result in weak muscles and weak transmission of the signal uh, back and forth across the neurotransmitter gap. Goodpasture syndrome. This is an antibody to the basement membrane and lining of the glomeruli and the lung alveoli. The immune complexes bind to basement membranes, and kudos to anybody who can tell me why it is a type 2 hypersensitivity and not a type 3 or 4. And the antiglomerular membrane antibodies are what's responsible. So, uh, autoimmunity results from the loss of central or peripheral tolerance. Remember that that's our way of weeding out all our autoantibodies, either at the central production where they're located for B and T cells, or where they antigenically mature. Exposure to environmental factors and injuries can trigger autoimmune diseases in susceptible individuals. And to summarize, we have the glomerular rheumatoid arthritis and systemic lupus as our major examples of systemic autoimmune disease. The ANA is the gold standard. You should recognize the patterns and know them. Again, we don't need to memorize every single antibody for systemic lupus, but to know in a general sense that in addition to double-stranded DNA antibodies, there are a host of other ones, and they can be referred to on the chart. You have the rheumatoid arthritis, which is directed against the FC portion of the IgG, and the glomerular disease that attacks the cytoplasmic granules, white blood cells, or granules. Organ-specific diseases that we went over are listed here, and we have the specific antibodies. So again, this is a good study chart for you, as it lists all the antibodies associated with the particular organ-specific pathology.